Okay, so this is the opening screencast for the part of the reproductive system, the second part. So we've gone the reproductive system's done, and now we're going on to the production of a cell or cell division, mitosis. So this, I believe, is screencast number 4 or 5. I've lost my count. 4 or 5. All right. And that would be mitosis. This one is just an opening, the opening slide. So there's about a few, but there's not tons and tons of notes on here. There's a set of definitions I do want you to have that I'll review with you. All right. So again, mitosis, as you know, is a requirement not only for you to develop into a person, but for many of your cells in order for you to produce any new cells. All right, so the fertilized egg is the first thing we always speak about. Starts off with one egg, 2 sets of DNA, okay, one from your mother, one from your father, and then division, division, division before what happens is called differentiation. All right, so it must divide. And why do cells divide? There are a few reasons. Well, we just said for reproduction, to reproduce a species. All right, one-celled organisms. Okay, asexual reproduction. Okay, they give it, so it doesn't matter if it's a one-celled organism, it is a clone, it's cloning itself. And asexual reproduction, and that is reproduction with one parent. All right, so for reproductive purposes, for growth and development of multicellular organisms. All right, don't love the noises. All right, so fertilized eggs, multicellular organisms. So these are the reasons behind why cells divide. And of course, for repair, if you cut yourself, okay? If you cut yourself, some organisms like the starfish you see there can regenerate their little spokes there. So reproduction, growth, and repair. Why do cells divide? All right, salamander also can grow a tail if that tail becomes removed. Okay? So there it is. Replace cells that die from normal wear and tear, like your skin cells, and also from injury. Okay, what has to be copied? Really everything in a cell. So we're going to go over the cell cycle. So really everything in a cell must be copied. Number one, of course, is DNA. And so if something goes wrong there, that could be a mutation. So, all right, dividing cell duplicate. I'm going to actually take the sound off because I'm annoyed. Goodbye. Okay, creates 2 copies of itself. And when you are dividing a cell into 2 cells, those must be identical. So mitosis, identical cells, 2 identical cells. All right, and we start to get used to some of the genetic terminology here. They call a daughter cell, sorry, there are no such thing as son cells. So a copy of its cells, everything copies inside a cell. That one cell splits into 2 cells. Those cells are called daughter cells, but they're actually parent cells of the next 2 daughter cells. All right, so you see, all right, what a cell normally looks like. Doesn't look like, the DNA doesn't look like what we think it does, the classic X look of the DNA. The DNA, in fact, only looks like that X when it is getting ready to divide. All right, then it becomes more organized, the organizing and packaging of DNA into chromosomes. That is a process that's called supercoiling. All right, so you can see here, chromosomes. These are chromosomes. Chromosomes generally live its lifespan in a cell as a single strand, not a double, not the X, a single strand. So in this cell here, you have 4 unique chromosomes, all right? 2 blue and 2 red chromosomes. There is a similarity between the blue and the red, but they're distinct chromosomes. So chromosome number 1, number 2, 3, and 4 within it, all right? An everyday working cell. So we get ready to divide. All right, now the reason why it's not loosely bound like this is because this DNA has started to thicken, to condense, to coil in it. All right, and so there are 4 chromosomes here. All right, so let's go. We're going to go to take a look at the DNA in just a second down here and actually label this picture here. All right, so most chromosomes then, you can see this process here that you see in this diagram is called supercoiling. The DNA wraps itself around a round protein. That protein is called a histone, H-I-S-T-O-N-E, T-O-N-E. That word's not incredibly important. It wraps around a protein. All right, so some of the most important words here. Again, histone, not the most important, but it's the process of the DNA wrapping itself around as it gets ready to. So we're going to define all of these words on the next page. Now, this diagram, I'm going to go to the smart board. Here's a diagram. Sorry, wrong one. Here's a diagram that I'm going to want you to draw into your topic 12 Cornell. Do not take this out, draw it in. All right, so you have your standard chromosome here. Okay, your standard chromosome here. So let's start on the bottom. So if you're looking at a chromosome, actually the chromosome has two parts. All right, and this is a replicating chromosome. So when it gets ready for mitosis, all the DNA replicates. So this portion of the chromosome, which is by you see the cursor there, that's one half, and this is the other half. And this is identical DNA material here. So this identical DNA material, so this was one strand and then it replicated itself, and we'll go over that replication process. So what is number one here? Number one is referred to as a chromatid. All right, so a chromosome contains two chromatid. And most chromosomes here do not have see the red dot right in the middle. It has either a short end or a long end. However, the red dot in the middle is called a centromere, M-E-R-E. And we'll get that all on one panel, and then we'll make it smaller here. A centromere, that is going to hold the two chromatid together. So each chromosome has two chromatid in the end. That's replicated chromosomes. All right. This, depending on where it is, this is simply called number 3 and 4. This is called the short, get another panel up here, excuse me, the short arm of the chromosome. Number 3, because it is a shorter amount of less genetic material. And I bet you can't just find what the bottom one is, the long arm of the chromosome. In short and long arm, things like that, not super important there. But we just want to make sure that you're aware of what these things are. All right, now the next page here has some definitions that I'm going to want you to have into your Cornell, referring to the structure of a chromosome. Oops, coming up right back there. They are. Sorry, I didn't have my smart board. I'm going to go through them with you. All right, this is kind of the hierarchy. So remember way back when, your cell, tissue, organ system, organism. This is the same kind of idea, except with genetic material. So what is DNA? DNA is nucleic acid found in cells, and that genetic material. Okay, so these are chemicals. We're going to go over the structure of the DNA molecule coming up in a future PowerPoint. That's molecular genetics. So we'll do this first. So chromatin, there are 3 C words that you need to know. Chromatin, chromatid with a D, and chromosome. So chromatin, all right, is the DNA in the protein, okay, DNA plus protein in what is called the G1, the S, and the G2 phases, and we'll go over that, so don't worry about that now, of the cell cycle. So basically, everything in the cell cycle except when the DNA is ready to undergo mitosis. All right, so the whole life cycle of the cell except for when it's ready to divide. What is a histone? Guess I did throw it in. That's the protein that contains the DNA wrapped around it. So you'll see. So a histone is a classification of protein. Again, it's not the biggest deal. All right, a chromosome contains hereditary units, the genes. Chromosomes, we usually use the word only when it is in the process of dividing. And again, IB is restricted from using anything but chromatin, chromosomes. They have to have it in order. So a chromosome, but we're a little bit more lax, especially in the regions. They use chromosome because their chromatin and chromatid are not part of regions. So chromosome is the general word that contains the hereditary units called your genes. A chromatid, okay, is named to a chromosome when there is identical copy made. So we're going to go back to that picture for just a second, then we'll come back to the diagram. So this is a chromosome. Chromosomes that are ready to divide contain 2 chromatid. They are which contain exactly the same genetic material because one of these chromatid is going to go into the new cell and the other is going to go into the other new cell. Okay, so the other cell, I don't know if it's new cell, the other cell. All right, so chromatid, when there's an identical copy made. Sister chromatid, two identical pieces of DNA. Go back to the picture again. Chromatid, chromatid. Hello, sisters. These are sister chromatid. They use a lot of female sir words in genetics, no male words. So a chromatid. All right, together, one and the other, sister chromatid. So there, whoops, back to the definition, sorry. All right, a centromere, that's the region to where the sister chromatid are attached to the chromosome. Sister chromatid attached to the chromosome. Now I'm going to take the mask off here. All right, so there are 6 or 7 words here that you need to be aware of. And again, the word chromosome is used mostly by the regions, but we're going to try to give you just a little bit more. All right, so let's go back to the PowerPoint. All right, and you can see an actual chromosome. Let's bring it up. An actual chromosome there. All right, so this is a chromosome 2 as well. However, this chromosome does not necessarily have to have the exact same DNA on it because this is your chromosome from one parent and this is your chromosome number 1 from the other parent. So you have 46 chromosomes in, but you have 2 identical chromosomes for each parent. You have 2 chromosome number 1s. You have 2 chromosome number 2s. All right, now let's take a look then further. So when this gets ready to divide, all chromosomes have to make an exact copy. So this here only pertains, let's say, to this chromosome. So the chromosome makes a copy of itself. So it has 2 identical sets, 2 joined chromatid. And although it doesn't show it, this one, if I could draw one, would have another X here. So let's say this chromosome only comes from this chromosome. One parent, the other parent. So it's actually better if I could have a pen here to draw. Maybe I can have a pen here and draw. All right, so you'll notice here, one parent chromosome, the other parent after division first starts out before division as a single-stranded chromosome. See it there? And before division, there's a part of the cell cycle that doubles all genetic materials. And right before mitosis begins, the chromosomes organize themselves. All right, so copies and packages. Copies the DNA, cells double up in a spool, and there you see an electron microgram of a chromosome, okay, right here, ready to divide. Doesn't come out that way. This process of copying or DNA replication we'll do in molecular genetics, how that happens, all right? So you can see those are really more talking notes than they are notes in and of themselves, all right? So there you see it. All right, the 4 chromosomes, 4 single-stranded chromosomes. We're going to copy, and now you see this is a cell ready for mitosis. Each one has its sister. So here's the mother chromosome, father chromosome blue, mother chromosome of number 1 red. 2, so 4 chromosomes, 2 identical, okay, 2 identical. But remember, if you go back here, you know, we start talking a little molecular genetics. This one has the same genes on it. But remember, you may have a different gene for, let's say, eye color on one gene than you have on the other gene, but one gene is dominant to the other. And that's going into Mendelian genetics. Too much for this one right now. All right? Duplicated chromosomes. Ready for mitosis right here. Anything before mitosis. And there they are in electron microgram. Okay?